Okay, so the third factor, or the third point really that I wanted to make is that there is some specificity. Trevor really highlighted this point, and I think it's important. We were interested in serotonin because superficially some of these detrimental effects of dopaminergic medication resembled those of serotonin depletion, and there's kind of an old literature about some of these side effects of medication perhaps reflecting serotonin depletion. So we asked whether these effects on reward versus punishment learning are also seen with manipulations of serotonin. Now, this question is not trivial because serotonin projects pretty much to the same regions as does dopamine. Not exactly, there's some specificity, but broadly speaking. And moreover, like dopamine, serotonin has long been associated or implicated in reinforcement learning and decision making, exactly those cognitive domains that we all think about when we think about dopamine. Unfortunately, unlike dopamine, there's not no similarly well-developed formal theoretical framework for interpreting and guiding empirical work. Perhaps the best or the most popular ideas for a while come from Peter Dehaan and Nathaniel Doe and colleagues who've revived older ideas, of course, by Bill Deakin and colleagues to suggest that serotonin serves roughly as a motivational opponent to dopamine. Where dopamine codes this reward prediction error, perhaps serotonin neurons might code for this punishment or aversive prediction error. That's what the essence of that motivational opponency story entails. We wanted to test that, and we decided to use these learning paradigms that we also used for this Parkinson work that I talked about, for the dopamine work. So in a first study that I want to tell you about, we used this probabilistic reversal learning paradigm, this instrumental task where people actually choose a stimulus and then get rewarded or punished based on their choice. This paradigm, well, I just described it to you earlier, but we performed it in the scanner, so this allowed us, and we had subjects reverse between the two objects multiple occasions, so we looked at reversal-related signal. To test this idea that low serotonin levels would be associated with poor punishment learning, which is what you would predict, right? If serotonin activity would code for this aversive prediction error. So we used an acute tryptophan depletion procedure, which is a dietary procedure. Basically, people get a drink, can also be administered in pills, but we use a drink which contains all large neutral amino acids except tryptophan. These amino acids, they compete at the blood-brain barrier, and tryptophan loses the competition. That's partly what accounts for the serotonin depletion centrally. You have to give the drink, it's kind of nasty, and then wait for like 5-6 hours for the drink to take effect. But then it's quite a robust manipulation in our hands. So we can do blood sampling and actually confirm that tryptophan levels, tryptophan being a precursor of serotonin, of course, is depleted, 6.5 hours in this case, compared with a placebo drink or a tryptophan balancing drink, which contains all amino acids, including tryptophan. Okay. We used this reversal paradigm and looked at bold signal, and what we see in this first study was a quite remarkable increase in bold signal as a result of serotonin depletion in brain regions quite strongly associated with punishment processing. So the dorsomedial prefrontal cortex, here at the top front of the brain, close to the anterior cingulate cortex, which of course has often been implicated in error and punishment processing. Also more lateral orbitofrontal cortex. These regions were potentiated. Signal in this region was potentiated at the time of punishment, when people receive punishment. So the valence specificity of this effect was consistent with the motivational opponency hypothesis, but the direction of the effect wasn't quite, because depleting it actually potentiated punishment responses. We next did a study using this kind of adapted reversal learning paradigm, which allowed us to disentangle reward and punishment learning to test much more directly this idea that tryptophan depletion would affect selectively punishment learning while leaving unaffected reward learning. Now, the first thing that I can say, I don't have a picture, is that the trial type that is consistently affected by dopamine manipulations in many, many different studies now, that's the switch trials, the trials after those unexpected outcomes, was unaltered by tryptophan depletion. So adapting behavior on this task is unaltered consistently, now in two different studies with tryptophan depletion. What was altered was performance on non-switch trials, on trials where people had to predict punishment. So throughout this task, people are presented with this face and this scene, and they have to indicate whether they expect punishment or they expect reward. On some trials, things reverse. They have to sort of pay attention and adapt their behavior, but on most trials, they just have to sort of implement their prediction. They go, okay, this was the punishment-associated stimulus, I predict punishment. This was the reward-associated stimulus, so I predict reward. What you see is that in the placebo, tryptophan balancing, the plus condition, the white bars, people are slightly worse at predicting punishment than at predicting reward. They have a bias away from wanting to say this is punishment. And that bias is abolished by tryptophan depletion. So lowering serotonin in the brain actually enhanced the ability to predict punishment. And this was not an isolated effect. This is also replicated in a completely different subject sample. Again, in that new study, different subjects, we see no effect on the switch trials, but a valence-specific effect on the non-switch trials. So experimental lowering of serotonin enhanced the impact of punishment on bold signal, that's what we saw in that first fMRI study, but also on prediction learning. So that's consistent with its implication of serotonin in clinical depression, but, one second, the open question still is how to reconcile that, of course, with theoretical modeling work, suggesting that these serotonin transients that lowering serotonin transients would attenuate an aversive prediction error. It's also not easily reconcilable with the implication of serotonin in impulsive aggression, which is also accompanied by low serotonin level and presumably reduced impacts of punishment on behavior. So there's a number of open questions, and I decided to focus the rest of this session on dopamine because I don't understand it yet, basically. So I don't really have a good answer. There's a number of hypotheses that we're working with to do with inhibition in interaction with aversion, but that again is a separate talk. And I think Zach might talk about some of this tomorrow. Yeah. I saw some fingers. Yeah?
Is the tryptophan depletion, so that was, I guess, is that chronic levels of serotonin in and like that?
Yeah, exactly. Yeah, so that was one of the hypotheses that we put forward. So one possibility, of course, is that with tryptophan depletion, what we are doing is moderately lowering sustained levels of serotonin in the brain, which then enhances the dynamic range for stimulus-evoked transients, punishment-evoked transients. One possibility. Not mutually exclusive from another possibility, where the effects of serotonin are best understood in terms of an interaction and intrinsic coupling between inhibition and aversion, something that Molly Crockett has worked on, and Peter Dayan and Quentin Huss and various other people. But again, yeah, that's a whole different story.
Excuse me.
Yeah.
And tryptophan depletion is affected on the neurobiological system.
Your question is whether some of these effects reflect manipulation of noradrenaline. Yeah. This particular paradigm we haven't used, this reward punishment paradigm we haven't used with noradrenaline manipulations. I would anticipate not to see valence-specific effects. That's my hypothesis, that you might see prediction error-like, modulation of prediction error-like responses, but possibly not as a function of valence. Yeah. Yeah? I would say that this pattern of effects of serotonin cannot easily be reconciled with the motivational opponency hypothesis. The story is probably more complicated than that. But what we can conclude is that effects of dopamine on learning, which, by the way, we cannot really disentangle from effects on choice, but that's an incidental remark, differ qualitatively from those of serotonin manipulations. Just highlighting the neurochemical specificity of these drug effects. Of course, showing the specificity of dopamine's effects in relation to noradrenaline is yet a different challenge, which we haven't quite embarked on. But okay, the last point I want to make. How much time do I have left?
Oh, good 50 minutes.
Oh, okay, good.
32 questions so far.
Yeah. No, that was already before the break. Oh, it's 30. Okay. Okay, so the last point I want to make is that there's multiple neurocognitive routes through which dopaminergic drugs can alter cognition. Of course, dopamine modulates multiple systems, multiple functions, and much of the literature focuses on this learning and choice domain, reward. I've talked about that so far. But I want to shift attention now to this whole other domain of cognitive control, because dopamine also has very remarkable pervasive effects on cognitive control and working memory. In fact, the first evidence for a role of dopamine outside of the motor domain was from Pat Goldman-Rakich in terms of working memory. And those effects, those cognitive effects, cognitive control effects of dopamine are generally associated with modulation of the prefrontal cortex. So Brzozowski and colleagues, Trevor referred to this, but let me just highlight that again, showed that 6-hydroxydopamine lesions in the prefrontal cortex impaired performance on the delayed response task to almost the same degree as the complete ablation of the prefrontal cortex. And that effect was remedied by administration of dopamine agonists and not by drugs affecting serotonin and noradrenaline, and I believe also the cholinergic system. Now that important role of dopamine in working memory, in particular this delayed response performance, was reinforced by neurophysiological work showing that application of D1 receptor agonists enhances delay period activity during when animals perform a delay response task, and antagonists of the D1 receptors disrupt delay period activity. Also, Williams and Goldman-Rakich in 1995 already showed that doses that are too high, excessive doses, abolish delay period activity. So that actually provided the basis of this inverted U-shaped relationship, which all started in the prefrontal cortex, and later we then extended that to the striatum and learning, but it was grounded in this prefrontal working memory work. And the idea is that excessive D1 receptor stimulation actually blocks input to prefrontal neurons, possibly by potentiating not just the glutamatergic inputs, but also the GABAergic interneuron activity input onto these extrapyramidal cells, as a result of which new input doesn't reach the prefrontal cortex any longer. This kind of work also provided the basis for the dual state theory of prefrontal dopamine by Dershowitz and Siemens, who state that optimal levels of D1 receptor stimulation in the prefrontal cortex are key for stabilization of current task-relevant representations in working memory, and that both too little and too much D1 receptor stimulation are associated with reduced signal-to-noise ratio and enhanced impact of noisy events on prefrontal working memory networks. We aim to assess somewhat indirectly this hypothesis and designed a paradigm that measures both the stabilization of working memory representations and also the flexible updating of these working memory representations in the context of new input. Because one implication of that idea is that supra and suboptimal levels of D1 of dopamine receptor stimulation in the prefrontal cortex would be bad for stabilization, but good for flexible updating of working memory in response to novel input. Huh? You're listening to me, and it's a good thing to be stable and to focus, but if now a fire burn breaks out, you better shift your attention and start to adapt your behavior. Here's a model of that situation. We have a simple delayed response task. People attend to the screen and are instructed that they have to memorize the shapes when they see a T in the middle of the screen. So here, the encoding stimuli, they all encode, and then there's two conditions. In one condition, people are presented with two novel intervening stimuli, and those stimuli are accompanied by a T. And they're instructed if they see a T, they better replace their old working memory with these new items. So they have to update, they have to use these new stimuli to update working memory. In the other condition, without a T, they have to ignore these stimuli. They're instructed to watch the screen, but to ignore the stimuli. And then at probe, they have to indicate whether this probe stimulus matches one of the two task-relevant encoding stimuli, which are different for the update and the ignore condition, right? So in terms of motor and sensory demands, these conditions are perfectly matched. The only thing is that in one case, they ignore the intervening stimulus. In the other case, they actually detect it. We were interested in this study in manipulating the catecholamine system more generally. We looked at the effects of methylphenidate. So we didn't here aim to disentangle effects of dopamine and noradrenaline. But of course, it's relevant in the context of this dopamine talk. What we see is remarkable. We see that administration of methylphenidate, a low dose, 20 milligrams, improves, consistent with its enhancing effects in ADHD, the ability to ignore intervening stimuli in the ignore condition. They perform better in the ignore condition. But those same subjects, the same drug in the same subjects had the opposite effects in the update condition. So now, actually, subjects on methylphenidate perform worse when they have to update, use this stimuli for updating. So methylphenidate potentiated stabilization of working memory representations, but actually impaired the flexible updating of those representations. And these effects were accompanied by opposite effects on bold signal in the prefrontal cortex, BA9 and also dorsomedial prefrontal cortex. So what we see is that administration of methylphenidate potentiated signal at the time of the intervening stimulus during the ignore condition, but attenuated it during the update condition.
So just keep methylphenidate, did you use for healthy volunteers?
Yeah, sorry, it's healthy volunteers, yeah. We did do a number of studies using more selective dopamine drugs, using a slightly different paradigm, which I won't bore you with, but this is a summary of that other work. So for example, we've shown that Parkinson's disease, off medication, associated with striatal dopamine depletion, among other things, is associated with cognitive inflexibility, but actually better cognitive stability. And now again, of course, Parkinson's disease is not that selective for dopamine, I should say, admit that. And also the effects of dopaminergic medication, though, were opposite on these two types of function. So dopaminergic medication, you can see that. Let me get this. On medication, patients with Parkinson's disease were more flexible, switch costs were reduced, but actually their distractor vulnerability was restored. So dopaminergic medication in Parkinson's disease had opposite effects, which might reflect a restoration of frontal upregulation of dopamine. We also see contrasting effects on flexibility and stability of bromocriptine in healthy volunteers. So what we've seen is that bromocriptine reduces switch costs, but actually enhances distractor costs. So bromocriptine made people more flexible but less stable. Okay, so this is reminiscent of some of the work, I guess, that Martin talked about. So I'd like to pursue some discussion about possible interactions here with the cholinergic system. What we've seen so far is that dopaminergic drugs like methylphenidate, levodopa, and dopamine receptor agonists improve or impair classic forms of cognitive control and associated prefrontal bold signal depending on the specific demands of the task under study. So it seemed to boost cognitive stabilization at the expense of cognitive flexibility. And the fMRI results suggest that these effects on working memory might be mediated by the prefrontal cortex. But, yes?
Did you look into oscillations in these studies?
No, but I'm working on that actually with Ole Jensen. So I don't really have that expertise, MEG, EEG expertise myself, but we're beginning some collaborations. I'm particularly interested in that because bold is quite difficult to measure during a delay. When there's nothing on the screen, usually delay period activity in prefrontal cortex is really hard to measure. So we're beginning to do that. And we have some data set actually as a function of methylphenidate. And we predict that probably distractor-induced disruption of delay period activity might be reduced, diminished as a function of dopamine receptor stimulation. We'll see. I'm interested in that. Yeah.
So the methylphenidate, the effect is on the stabilization of pain is really great. I just wonder about that. Have you done any distinguish to make sure that this effect is because of the dopamine or neuroimaging?
Yeah. We have not yet in that study, but we're doing a study as we speak, and Lieke will tell you about that, to start to look at exactly that. So we're comparing effects with sulpiride, which is more selective. But we cannot, from these data, say definitively that this is dopaminergic. We do see effects as a function of levodopa and bromocriptine, but again, those are also not definitive. Yeah. Mm-hmm.
In the study where they had to update the shapes based on whether there was a T in the center,
Yeah.
is there a particular reason why such complex shapes were chosen?
No, there was no actually, and right now we're using this task with the kind of color wheel setup that other people, Luck and Zhang, have used. So the principle holds, irrespective of the specific stimuli. Yeah.
And do you think the fact that there were different colors also in the shape, do you think that is going to affect the testing?
Yeah, no, I don't think so, because we see the same kind of pattern with other types of stimuli. The reason Sean chose to use these particular stimuli is because he saw that it potentiated working memory. He had reasons to think that, changing the colors with the shapes, but there's no extra dimensional aspect here. There's no set element. It was just all congruent. Yeah. Yeah. Okay, so this fMRI data suggested that some of these effects of on cognitive control might be mediated by the prefrontal cortex. But, and now I want to get to this point, is that there are multiple routes through which these drugs can alter cognition. Of course, we know that cognitive control is an output, if you will, of functional modules interacting in frontostriatal circuits. So dopamine might well alter prefrontal function indirectly by acting on the basal ganglia. So indeed, in other work that I'm just going to sort of refer you to, but not going to detail, we've shown that the basal ganglia and dopaminergic modulation of the basal ganglia can gate or select frontal actions by modulating top-down connections from the prefrontal cortex to posterior sensory cortex. And this basal ganglia modulation of these top-down connections was associated with attentional shifting. Now, from the first part of my lecture, we've seen that one key function of dopamine in the basal ganglia is the learning and selection of actions based on reward and punishment. And this role of dopamine in the basal ganglia has been proposed to extend to the learning and selection of more cognitive actions, more cognitive task rules maintained in working memory. So this whole story of learning and gating of actions, simple actions, might extend to learning and gating of more complex cognitive tasks. And this then leads me to begin to integrate dopamine's role on reward-based learning and choice on the one hand, and in cognitive control on the other. And this then really leads us to begin to understand our motivation for cognitive control, or our willingness to exert control, which I think is one of the most fascinating questions. How do we motivate ourselves to exert cognitive control? Why are all of us spending so much cognitive effort to try and understand something, whereas other people might ask, why would you make your life so difficult by asking yourself all these questions? Now, the working hypothesis, so I don't really have an answer, but the hypothesis we're working with is that striatal dopamine might alter cognitive control indirectly by modulating value-based learning and choice processes that affect our motivation for control. So effects of dopamine-related disorders and their treatment on cognitive control might reflect, in part, effects on the willingness in addition to the ability to exert control. I've put rather than, but I should put in addition to the ability to exert control. One way to measure the motivation for control is very simple. So here we've used a very classic cognitive control task, Stroop targets. I guess you know all that. Huh? So people are presented with arrow word targets. In this case, they always have to respond to the word and ignore the arrow. The arrow is quite a prepotent stimulus, so they have to press the left button here. The targets are always incongruent, right? So they have to exert some cognitive control to overcome this irrelevant stimulus. And critically, this target was preceded by a reward cue. And the reward cue indicated to the subject whether they could win a lot of money or very little money. It's all relative, 15 cents and 1 cent. And so a measure of motivated cognitive control was the difference between high and low reward trials. Now we were interested in the degree to which this effect of motivation on cognitive control varies with individual differences in dopamine synthesis capacity. So we made use of this same technique I mentioned before, so FMT PET, so we measured dopamine synthesis capacity in the striatal terminals, and found a lot of individual variability in the degree to which this reward cue potentiated cognitive control. So here on the y-axis we see the effect of this reward cue on Stroop interference control, so that high scores represent motivational enhancement of interference control, and low scores represent actually reward-induced impairment of cognitive control. And interestingly, this variability was accounted for by dopamine synthesis capacity in the striatum. So that low dopamine synthesis capacity subjects, they benefited from the promise of a bonus, from the promise of reward, whereas high dopamine subjects, they actually got worse when they anticipated to be rewarded. Reminiscent of the overdose hypothesis, yes.
Is this a difference in performance on the two types of trials? I mean, what's the
Yes.
what's the effect
Yeah. Sorry, it's incongruent. I said this wrong. I said there was only incongruent trials, but there's incongruent trials and there's congruent trials, and interference control represents the difference between incongruent and congruent trials.
Also, still the effects that you're planning here is a different. So it's a difference between the congruent and incongruent trials.
Yeah. So if you take performance or error rate on incongruent trials, where the word and the arrow point in opposite directions, you subtract from that congruent trials. Then you have an index of cognitive control. Then you compare that in the high reward and the low reward condition. Here, high scores represent benefit. If you anticipate reward, you think, oh, I'm going to get a reward for good performance, right? So I better pay, try hard. And that helps. Yeah? It helps here in the low dopamine subjects, but it hurts the high dopamine subjects. Yeah.
This might be a very naive question. I don't know a lot about human studies. But do you see at point saturation, or it would be habituation in animal studies for these motivational tasks, that at some point it's not really relevant anymore because they have already received multiple rewards or are not interested?
Yeah, we don't. That's interesting. We don't see that sequential effects here. Of course, the reward is very low. They don't get it immediately. They're accumulating it. It's like nominal reward in a sense, and people just want to get as much money as possible. It's quite different from working with primary reinforcement. We've also tried to work with primary reinforcement, like juices, and then you get that kind of effect.
Would you expect if there were sort of like a saturation of motivation that you would have the same dopamine level, or what would be primary to that?
You are asking whether saturation is accompanied by reduced changes in dopamine? Maybe, I don't know. The assumption here is that dopamine mediates the motivational effects, and if people are saturated and don't care anymore, you might not see that. Yeah. Yeah.
Maybe my question is very simple, but I don't know why you chose the codate clues.
Oh, no. So I didn't look selectively in the caudate nucleus. So we do actually. So signal is high in the entire stratum, and it was highest in that caudate nucleus, which is really interesting because we also did the same paradigm with fMRI and saw that reward effects on this interference control was accompanied by bold signal in exactly the same region. So there were just no effects in other parts of the stratum. Yeah, I didn't highlight that selectivity. Yeah.
So would it mean that the higher dopamine, in subjects with higher dopamine, the linear range of motivation that's available is less? So that's why they are doing much better than what they start off with?
Yes.
People with low dopamine have a higher range of motivation?
Yeah. So the answer you trigger in me is that I think that's what you're asking, is whether there's more range in performance to be modulated or less. This cannot account for this effect. So there's no correlation between dopamine synthesis capacity and the interference control at low reward, for example. That also was not the case in the subsequent study, where we found the same thing in Parkinson patients. So I'm going to show you that next. So being motivated does not necessarily translate into greater cognitive control. Cognitive control can actually be impaired by reward motivation, depending on individual differences in baseline levels of striatal dopamine. This, by the way, this finding of choking, if you will, is not that new. So several other people had shown that. What this study added to that is the mechanism, a potential mechanism underlying this apparent choking under reward. So the next study I want to show you is essentially conceptually the same, but what we did was to look at Parkinson's disease as a function of striatal dopamine depletion. And we asked whether depletion of striatal dopamine in patients is associated with, paradoxically, enhanced motivation for cognitive control. That's what would follow from that previous study. Yeah.
Did you also look into, instead of different size of reward, into different size of punishments?
No, not yet. Interesting. We want to do that. Whether punishment has the same effect or actually the opposite. It's an interesting question. Okay, so here we had SPECT scans available in patients with Parkinson's disease. So this was a FP-CYT binding study. FP-CYT binds to dopamine transporter, so it's an index of presynaptic cell integrity. And it's often used also clinically, actually, to index Parkinson's disease. So you see a much reduced binding of CYT in Parkinson's disease relative to controls. Now what we find is that patients with Parkinson's disease indeed exhibit greater beneficial effect of reward promise on cognitive control. So dopamine depletion here seemed, or at least Parkinson's disease, from this plot we could only conclude Parkinson's disease is accompanied by enhanced effect of motivation on cognitive control.
So is it just so how long does the motivation take?
How long does the motivation?
Yes.
No, this is trial by trial. So we actually on each trial say, now you can get 15 cents for good performance. On a different trial we say, now you can get only 1 cent. And we subtract performance on the two trials and we look, when they expect to get a lot of more money, do they perform better? Or what we see here also in the older controls, when they expect a lot of money, they actually get worse. It's like, ugh, this is really important, so I screw up, kind of thing. We see that this effect is a function, quite strongly, of dopamine transporter binding in the striatum. So the more dopamine patients have, the greater the detrimental effects of reward motivation on cognitive control. Depletion here, denervation, is associated with enhanced motivation for control. Rather paradoxical, but now replicated in 3 studies. It's not a function of baseline performance under low reward. It also is reminiscent to me of paradoxical kinesia. So what we see in Parkinson patients is that there's difficulty with intrinsic motivation, motivating themselves to start walking and to stop walking. But when you provide a cue, this can actually help overcome an external cue. This can overcome some of their motor and perhaps cognitive deficits. Yeah.
So actually, you sort of semi-answered the question that I just had in my head, but you know, a lot of this you're saying stuff kind of says the opposite of what you're saying here, right? Which is essentially that those who are depleted in dopamine in Parkinson's disease have less motivation.
Yeah.
But I guess what you're saying, if I understand correctly, is that you're saying motivation for cognitive control is quite a specific case or quite different kind of motivation?
Well, I think the key difference here is whether we're looking at intrinsic motivation or extrinsic motivation. So this is incentivization. This is the effect of an incentive cue on performance. It's like, here's a cue and now perform better. This might actually trigger some system that is rather different from the type of system that you measure with intrinsic motivation. So where Lieke actually is going to work with this kind of more intrinsic motivation paradigm to actually quantify the subjective value that people assign to cognitive control using preference tools. And now there's no incentive cue to motivate people, but rather we quantify the value that people assign. And what we see Masoud Hussain and colleagues have seen in Parkinson patients is that people with Parkinson's disease assign less value to motor effort, but we might think that also cognitive effort value is lower. But that's rather different from the incentivization of cognition.
So I've got a question. Something you haven't mentioned yet, Roshan, which is important. I don't understand the significance. That is that striatal dopamine is often reciprocally related to frontal dopamine.
Yeah.
So in normal animals or normal people, the more striatal dopamine you have, the less frontal dopamine you have. It may be to do with the regulatory functions of prefrontal cortex on these midbrain systems.
Yeah.
Now, Supposing that's the case here, we have enhanced straight dopamine synthesis capacity, therefore reduced frontal synthesis capacity, associated with less motivation. So could the cognitive motivation effect exerted on cognitive control be mediated by frontal dopamine?
Yeah.
You get it?
It's also, so this neurochemical reciprocity is shown, for example, by Angela Roberts, right, where they, well, you were involved with this, do striatal dopamine, 6-hydroxydopamine lesions in the frontal cortex and see upregulation of striatal dopamine levels. In Parkinson patients, there is some evidence for upregulation of dopamine synthesis capacity. There's a poster here, someone was just mentioning this. It's also the case in thalamus. So this is possible. We don't know. We don't know. So this is an empirical observation, replicated multiple times. I strongly believe that the motivation, incentive motivational effects on cognitive control are potentiated. And we can, of course, use that, right, to exploit it in the approach of patients. But
yeah, it's more or less linked, basically. I was wondering about the prefrontal inhibitory control and norepinephrine, along with these kind of results in Parkinson's disease.
Yeah, I think it's a very interesting idea, right? And so one thing that I've been wanting to do, but I haven't gotten around to, is to actually look at effects of cortical stimulation as a function of pre-treatment with dopamine agonists or antagonists to get at that frontal control mechanism and assess whether there's a role for the prefrontal cortex in controlling these subcortical dopamine effects in cognition.
Yeah, the question about prefrontal dopamine release, noradrenaline release in prefrontal cortex against the limbic release.
Yeah. I think James will talk about noradrenaline tomorrow in Parkinson's disease, Wednesday. Yeah.
So the prediction would be in severe Parkinson's, the effect would collapse because all dopamine would be, in a sense. At least my prediction.
Yeah. In your case, that's a prediction we could test. No, so there was someone there first. Yeah.
You haven't shown it when you presented the task, but I assume they get external feedback, so they get the high and low reward cues, they have this group, but then they also have immediately the feedback if they're correct or incorrect. Do you think you would find different results with only internal feedback, like giving away the external feedback?
You mean so sequential effects as a function of outcome on the previous trial? We've looked at that, yes, and there's no effect. Yeah. Yeah, well, that would be error-based slowing or these kind of Gretton-like effects. And we don't see that here. Of course, there's now a cue in the middle. So it isn't really set up perfectly, this design, to look at those sequential effects.
I was also thinking about confidence, actually. Like this internal feeling, I'm correct. Now I'm correct. So that actually would explain it also. That they need actually the immediate feedback to improve.
I doubt it. We don't know. I can't say, but I doubt it. I think it's rather this incentive, this extrinsic nature of the motivation that's important. But it's an open question. Yeah.
Did you look into the response time to not necessarily this one, but the one you showed before with the delay? Like how long did the response time work from the indexing image was shown until it responded? Or did they not have to respond?
You're asking whether these effects are seen also in terms of latency? Yeah, no, there's no effects. These are accuracy effects.
Do you take into account their baseline response time? So you just like have that as their 0 and then calculate the response time from that? I don't know if you...
So this is error rates, right? But and so what we are sure is that if we correct for baseline error costs, so this is not a function of there not being dynamic range, for example. We haven't also taken into account changes in reaction time, but there weren't any clear effects there. Okay. Yeah. On reaction time.
Do they answer verbally or like motor skills?
No. No, it's responses.
Okay, so...
It's button box.
Okay.
In this case, yeah. Yeah. Okay. Reward motivation potentiates cognitive control in low dopamine subjects. It's stating low impulsive here as well. I'm not showing you that data. Accidentally left this in. But impairs it in high dopamine subjects. And these findings raise the hypothesis that at least some of the effects of dopamine-related disorders and their treatment on cognitive control might reflect, in part, reduced willingness rather than ability to exert control. I think that's pretty relevant given that, for example, dopamine synthesis capacity is enhanced in various impulse control or disorders accompanied by impulse control deficits like pathological gambling addiction. For example, we've just shown that that is accompanied by enhanced striatal dopamine synthesis capacity. The next question we asked is whether effects of methylphenidate on cognitive control, the ones I presented just earlier, might also reflect modulation of the willingness to exert control. So here we perform a very large study on the effects of methylphenidate. And we're interested, by the way, in methylphenidate not for its neurochemical specificity, obviously, but for its common use. Many people are using methylphenidate, also healthy volunteers, increasingly so, students and academics. I know colleagues who have a little Ritalin pill in their pocket when they go to conferences. So we're interested in those effects.
But not the present faculty.
Yeah, is that right? I don't know. Oh no, you were optimized already. Yeah. In this study, we included just over 100 subjects in order to take into account individual variation. We were unable in this study to measure, to obtain PET scans. So as in this previous study, we took into account baseline working memory capacity as a proxy of dopamine. And we employed in this study a demand avoidance, a cognitive control avoidance task developed by Matt Botvinnik and Wouter Kool and colleagues in order to assess the degree to which people learn to avoid cognitive control, and the degree to which people are willing to engage in cognitive control, in addition to being able to exert cognitive control. So in each trial here, they choose between two abstract patterns. One of them is associated with a difficult paradigm, a difficult task. Basically, they have to switch between tasks a lot if they choose this one. And the other pattern is associated with an easier task, a task repetition paradigm. So basically, it goes like this. They choose between the stimuli. Then they get one digit, which is either yellow or blue. If it's yellow, they decide whether the digit is odd or even. If the digit is blue, they decide whether it is higher or lower than 5. On each trial, they choose and they see a digit. If they choose the difficult task, they are confronted with task switching on 90% of trials. If they choose the easy task, they have to repeat the task on 90% of trials.
So after you choose this, you have a number of trials in which you can switch or not?
No. So basically they choose, they have one digit, then they choose again, and then the digit is a switch or a repeat trial relative to the, so it's very slow switching. Nevertheless, people avoid the task significantly. So it's not a very large effect, but overall, people avoid the task.
So, on this case, the right pattern is a difficult one, right?
Yeah, I pointed to the wrong one. Eh? Is that confusing? Yeah, all the texts. Okay, so on this next slide, I'm showing you the effect of methylphenidate on demand avoidance. So it's the proportion of low demand choices. Lots of individual variability, not surprisingly, but we could account for some of this variability as a function significantly of working memory capacity. And interestingly, this is a pattern that we saw on each of 6 paradigms included in this particular study. So I'm quite confident in the robustness of this span-dependent effect of methylphenidate. And what we see is that the greater the working memory capacity, the more methylphenidate increased demand avoidance, the more people were willing to avoid cognitive effort when taking methylphenidate. So this is the data split up in the high and the low span subjects. What you can see is in the high span subjects, methylphenidate promoted demand avoidance. In the low span subjects, if anything, it reduced demand avoidance. Now you might ask, well, is this an indirect consequence of them performing worse? Of course, if you perform worse, you would not like to engage in it very much. It is not. If we look at the performance, so that's basically the difference in performance on the switch task and the performance on the non-switch task, we again see a span-dependent effect, but in the opposite direction. So the more subjects' working memory capacity, the more methylphenidate reduced the switch cost, so improved performance. So methylphenidate in high-span subjects enhanced performance, but also motivated people to avoid it, avoid the difficult task.
Are they receiving rewards for any of these tests? For this test?
No.
Okay, so the only variable is the cost and time?
Yeah.
Okay.
Yeah, the origin of this avoidance is a subject of intense study at the moment. What makes people avoid cognitive control? Is it error likelihood? The effect of methylphenidate cannot be accounted for by error likelihood. Effects can also not be accounted for by sloth, by more time, because actually the effect on performance was the opposite. Is there something intrinsic about cognitive control that's aversive? We think so. But this is currently under study. Yeah?
Yeah, it comes to what you're just saying. What can you know about the behavioral specificity of this? I mean, are you sure that it's dopamine dependent?
No, and this is exactly why we're now engaging in a subsequent large study with methylphenidate. If I have time, I can tell you, because I wanted to show you some of the more practical aspects of running a study like this, if we have time. And then I also explain to you the rationale for that current ongoing study, which is to get at the neurochemical specificity, at the neural mechanism, so fMRI as well, and various other parts I can explain to you. But currently this is the first step. What it shows is that in high-span subjects, methylphenidate improves cognitive control performance, but also reduces motivation to exert control. So these findings raise the hypothesis that at least some effects of dopamine-related disorders and their treatment on cognitive control reflect reduced willingness rather than impaired ability to exert control. So there are multiple routes through which dopaminergic drugs, or catecholaminergic drugs, I should be precise, can alter cognition. They might reflect modulation of value-based learning or meta-decision making about whether or not to exert control, as well as the ability to exert control. And that leads me to this final sort of slide of this aspect. 4 principles of neuromodulation by dopaminergic drugs. The first one, baseline dependency. We've seen that in the Parkinson study, the overdose hypothesis. We've also seen it in the PET study with bromocriptine. The task dependency. Reward and punishment learning have distinct neural signatures characterized by differential optimal levels of dopamine. Reward learning is optimized with higher levels of dopamine than is punishment learning. Stabilization of working memory is optimized with higher levels of prefrontal dopamine than is updating of working memory. I've illustrated neurochemical specificity using exactly the same paradigms, where we see that effects of dopamine do not resemble, they also don't mirror the effects of serotonin. And I've illustrated this point of multiplexing, where there's multiple routes through which drugs might alter cognition.
Yeah, just a question about the last experiment with methylphenidate.
Yeah.
I was wondering if the methylphenidate could enhance or be related to fight or flight response.
Fight or flight?
Response in terms of
Fight or flight. Let's see. So we ran 6 experiments actually in this methylphenidate study, and one speaks somewhat to this fight-flight response. Here we looked at basically approach avoidance type of decisions based on reward or punishment in a crossover design. And we see that methylphenidate potentiates the Pavlovian control of behavior. That's mediated by modulation of Pavlovian responding, but also by a diffusion of credit assignment. Because what we think is that methylphenidate prolongs catecholamine transmission, thereby interfering in a sense with credit assignment. So with the assignment of reward to an action. And what you see is a potentiation of all actions leading to a general go bias. So that might address this question that you have, but it doesn't really follow from the work I've shown here. Because the demand avoidance is really about very subtle difference between task switching and task repetition, right? A general sort of fight-flight reaction would not really speak to the sophisticated preference for a slightly more cognitively difficult task, but not motorically difficult task.
That's because they were more focused, but also more motivated to avoid. So we're thinking maybe the more intense the task, the more avoiding the stressful task, the stress.
Yeah, it's possible. It's possible. So that actually they were exerting more effort, and that's why they perform better, and that's aversive. Yeah. So the cost of effort is greater. That's actually what we're assessing in the next study, where we quantify the subjective cost of control of cognitive effort. And we're predicting that it's this subjective cost of effort that's altered. Yeah. Yeah.
Thank you for your analysis. And do you have any idea about the molecular mechanisms which are responsible for this interesting observation?
Molecular, yeah.
The balance between D1 and D2.
Yeah. So what we would like to test in ongoing study is this idea that it reflects a modulation of, or a potentiation of D1 mediated go pathway activity, in which basically people are with methylphenidate or with D1 receptor stimulation in striatum better at learning from effort relief in this particular context. So what we're working on now is setting up an experiment where we modulate the reward punishment learning task, where we reward and punish people not with the actual negative positive outcome that we do, like the smiley face and the cryy face, but with a cognitive task that's either more or less difficult. So now we ask people to predict whether they're going to have to do a really difficult task or they're going to have to do a really easy task, and then allow them to basically learn these associations and update these associations based on unexpected cognitive effort or unexpected cognitive relief, and assess whether that is, you see these opposite effects as a function of dopamine.
And have you ever used this observation in clinical to treatment depletion?
No, not yet. No, we're not there yet. Yeah, that's something we'd like to do, but yeah.
Just pushing your questions up to 66.
Yeah.
I'm sure you've thought of this, but have you thought of comparing these effects with atomopsity?
Yeah, no.
Because,
So, yeah.
As I mentioned,
Yeah.
this is a very good control group for Ritalin.
Yeah.
Because it simulates what Ritalin
Yeah.
does in the cortex.
Yeah.
with no subcortical dopamine
That's right.
activity.
That's right.
So, it's an interesting
Yeah.
one. Would atomoxetine enhance cognitive
Yeah.
control?
Yeah.
Do you think
Yeah.
it might?
Yeah, I'd like to do that. I can tell you now what we're doing now. We had so many aims, and atomoxetine was one choice we considered. And in the end, we decided in this ongoing study to compare the effects of methylphenidate with sulpiride to get at the dopamine receptor selectivity. Also, given the previous data that I've shown you, where we see these quite selective effects of sulpiride on the striatum. So that was the first step. The obvious, if this is powerful and turns out to pay off, we have to compare it with atomoxetine. Is there time? A bit of time?
Still 4 or 5 minutes.
Yes, you've got
4 or 5? Oh, okay. Don't worry. Yeah.
So I'd like to ask a general question, and maybe it's not a good question, but let me try it anyway. So the cognitive neuroscience of dopamine function has enormously evolved, right? I mean, it's a very sophisticated science that has been formalized to a degree that doesn't happen in with respect to other neuromodulator systems and so forth. What has always frustrated me is that I'm missing the ability to take all the things you have learned and translate this into my understanding. Why do we see what we see when we lose dopamine or when dopamine works over time like in psychosis?
Yeah.
So the relationship between these sophisticated conclusions and the clinical symptoms that are associated with abnormal dopamine levels.
Yes.
I mean, you touched once a little bit at it, but really, do we have not more to say than it's in Parkinson's we have a low level of motivation for movement, and how this translates into psychosis, who knows anyway. So I guess what I'm trying to suggest or wonder is, can you hang your head out of the window and speculate a little bit more about how the conclusions from your research and from your field begin to give us a little bit more substance as to how to explain symptoms.
Yeah. Well, so I think actually Parkinson's disease is a really good example of where this insight is very relevant. That is a disorder, a prototypical disorder of the will. And in that sense, I think many of the cognitive deficits can be accounted for by a motivational deficit. And I do think that this insight then advances our understanding of Parkinson's disease considerably, where so far people focus on the motor symptoms.
Yes, but how about the motor symptoms then? I mean,
I see the motor deficits also as a motivational impairment, an inability to value the effort associated with motor activity. Some of it, of course, there's these other aspects, these cholinergic, and I've written with Trevor about this dual syndrome, and focusing on the dopamine aspects of disease. I do think that this motivational, this will deficit is core to the disease, and might well also be to ADHD. To me, that seems really, really clinically relevant. If we think about ADHD classically, we see ADHD as an inability to focus. People are so distracted. But if we reconceptualize ADHD instead as an inability to assign value to cognitive effort, then the approach to the disorder becomes entirely different. If it becomes a decision problem, and something that's what I believe speculatively reflects enhanced opportunity cost of cognitive control. The cognitive control is just too opportunity costly. There's just too many other valuable things around for me to focus. I just don't think it's a good idea to focus on this task because I miss out on playing with my kids and doing this other task, and I'm not going to finish this task because that really triggers an entirely different approach to this disorder than saying, okay, I'm going to train you to resist this distractor. You can train them endlessly. If it's not valuable for the patient, they won't invest in it. I don't think that's the problem.
What we need to do is to be comparing the effects you described with cholinergic modulation with these, because some of the concepts, while not identical, overlap in ways which we don't altogether understand, because we're using different paradigms which are subtly recruiting different
Yeah.
processes, probably. But there is obvious overlap.
Absolutely.
Yeah. I mean, the update ignore aspects of this work really overlaps with your paradigms. I think it's lots of potential.
Question here, 68.
Considering that it's involved so much in movement, have you looked into like differentiating between tremor and non-tremor PD patients to see if they respond differently on your different tasks?
Yeah, it's interesting. I'm involved with a study where we compare people with tremor, without tremor, and also dopamine responsive tremor and non-tremor. So like a factorial design to get at that. And we're using some of the same paradigms, neurocognitive paradigms, to look at that. Yeah, that's an interesting open question. Yeah, looking at GABA, for example.
The tremor patients, I think, traditionally have less dementia.
Yeah, exactly. Yeah, my intuition. Yeah. Although, of course, the implication also is that if you're well functioning, you're going to take Ritalin. You better not take it during studying because you're now no longer motivated to actually exert effort. You might perform better during an exam. I don't recommend it, by the way, but.
Okay, I think we better stop there. So,
Oh,
thank you very much.